Okay, so I will be starting right now. So today we're going to be learning about cellular communication and cell division. So there's going to be some pretty interesting topics today. Give me a quick second. Okay. Okay, so the first topic we're going to look at is the cell life cycle. So we've explored this in previous classes, but all of us are made up of cells, and we explored how cells work, the certain organelles in cells. Now we're going to explore the entire life cycle of the cell from its creation to its reproductive phases and to, well, the cells don't really die necessarily, but from like its entire life cycle. So, okay, sorry, my bad. So the overall life cycle can be summarized in this diagram. So there's things called the G1 phase, which is the first phase of a cell. And then after that, it'll go through the S phase, which stands for synthesis. And then after that, it'll go through the G2 phase, and G stands for gap. And then it'll go through the mitosis stage. And so basically, just a very brief summary. In this phase, the cell kind of grows. And then in this phase, its DNA replicates. And then in this phase, it gets ready to divide. And then when it its dividing process is called mitosis. So that's why cells don't really die. They just split into two different cells. And so the first stage we're going to look at is the G1 phase of the cell. So the G1 phase of the cell is where the cell grows, it makes protein, and it preps for DNA replication in the S phase. And it also acts as a checkpoint to ensure no DNA damage is done, because when you replicate DNA, you don't want to replicate the wrong DNA, because that causes many bad outcomes. For example, cancer cells, which we might explore in future lessons. And the job. So this is the phase of the cell where your cell does like the intended function to serve the organism. Like, for example, this is the phase in which your liver cells are putting out the hormones that you need to function, or this is the time where your skin cells are acting to help protect you. And the G1 phase is also primarily where cell growth happens, and cells almost double in size as they prepare for division. And so I like to go over DNA proofreading. So in every 100,000 DNA base pairs replicated, and we'll explore DNA replication in a couple slides forward, one of them is likely wrong. And this is actually a very high percentage because there's like millions and millions of DNA base pairs that are being replicated uh, in per cell, and you have like trillions of cells in your body. So that's a lot of error in this DNA replication process. So how your body proofreads it is through a protein called DNA polymerase, and this is actually the protein that builds new DNA, but it also can fact check it to make sure that it's correct. And remember, DNA, there was like these base pairs. Remember, adenine, which is A, pairs with thymine, which is T, and then the guanine in cytosine. And basically, if something is wrong, then this protein will be able to recognize that. Okay, so next we'll talk about the G0 phase of the cell. So the G0 phase of the cell is essentially where the cell kind of chills and stops dividing. And as you can see in this diagram, the G0 phase cell kind of shifts in and out between the G1 phase of the cell, which we explored last slide. And this G0 phase of the cell is where the cells rests, and they also do like the uh, intended function of your body. And this is like the cycle which you can stay in for like some cells, it's your entire lifespan as well. This is the kind of the cycle that it goes through if it doesn't like need to divide, and it probably shouldn't divide. Like for example, your neurons, which is your brain cells, you call them, they stay with you for life. They don't really divide because you don't want neurons constantly like disappearing and then they lose their function and then they grow back and gain their function, right? You want them to be in function throughout your entire life cycle. So these neurons, they only grow when you're like an infant or when you're in when you're in your mother's stomach and you're growing. And once your brain matures, they just enter this G0 phase. So they don't go through this other phase from the G1 cycle to like this mitosis phase. They just stay there because they don't need to replicate, and they will serve as your brain cells. And another great example of G0 phasing, like switching in between the G0 phase and the G1 phase, is liver cells. So, for example, you have your liver cells and they're doing their intended functions to serve your body in the G0 phase, right? Because they don't really need to replicate. But then say your liver gets injured, right? There's a scratch, and then the damaged tissue will release a hormone called HGF. You guys don't need to know this, but essentially it just triggers the sorry. It just triggers the cells surrounding this damaged tissue to actually switch into the G1 phase, so then they can regenerate via mitosis, which we'll explore later as well. The next we'll move on to the S phase of the cell. So the S phase of the cell stands for synthesis, and this is where DNA replication occurs. So in order for one cell to split into two cells, you need two copies of DNA in order to do that. So this is done through the S phase of the cell, and that's where chromosomes condense and they duplicate into sister chromatids. So this would be a chromosome, and then when it duplicates, it becomes like kind of two of them, and they're connected as something called a centromere. And then these, oh, sorry. My bad, guys. Can you guys still see my screen? Okay, hopefully you guys can. Um, and then, so these are called chromatids, and this entire thing would be called a chromosome. And it also acts as another checkpoint to ensure the DNA is copied without errors. And then finally, we reach the G2 phase of the cell. So this prepares cell structures and resources required for mitosis. For example, it will duplicate the mitochondria, or the chloroplast, or any other organelle, so that ensures those two copies of the cell later on receive sufficient organelles to function and to grow in the G1 phase. And also, yeah, it synthesizes essential proteins, and also again checks DNA integrity and make sure that nothing's wrong. Because and this DNA checking process happens throughout all phases of the cell, because it's such an important aspect. And if something goes wrong, it could cause cancer, which I believe any of you guys probably heard of it. It's not very good for you guys. So next, I just like to talk about DNA replication. So this happens in the S phase of the cell. I know this looks a bit like crowded and complicated, but it's actually one of my favorite parts in biology. It's extremely interesting to see how all these like different proteins work together to replicate DNA. So I'd like to first go through the list of proteins that take place in this DNA replication process. And the first protein is called helicase, and its function is just to unwind the DNA strand. So remember, the DNA strand is double stranded, so its DNA is like connected by base pairs. And so this helicase, as you can see here, helps unwind, like basically kind of unzip that DNA, so then that it can be so that these two strands can be act as template strands to duplicate. And then we have things called single strand binding proteins. And this function is just to bind to and stabilize single stranded DNA until it's used as a template. And this just helps stabilize everything and make sure reactions occur only when necessary. The next protein we're going to talk about is topimerase. And this protein, we usually don't see it in diagrams, but it would be ahead of the helicase. So it helps by overwinding strain ahead of the replication forks by breaking, swiveling, and rejoining DNA strands. So essentially, DNA is not always a straight line. It's like curled up into histones and stuff like that, like we explored last class. And this protein, topimerase, just helps like unwind that and prepare for helicase to come through. And then the next protein is called primase. And this is the synthesizer of an RNA primer at the 5' end of the leading strand and at the 5' end of Ozaki fragments. And Ozaki fragments is just how Okay, there's something called the leading strand and the lagging strand of DNA replication. We'll get to that in the next slide. But essentially, the leading strand can just be synthesized very quickly in one direction, and we'll explain why. And then the lagging strand has to be synthesized in these things called Ozaki fragments. And one really important thing is that the protein, which is the DNA polymerase 3, is the protein that actually adds these DNA nucleotide bases onto the template strand. So this is DNA polymerase 3, and this is the one that adds these bases, but it cannot add on to just a template. It needs a kind of like a starting point, and that's what this primase does. It sets this primer of RNA, so then the DNA polymerase can attach to it and then build off it. And then we have something called the DNA polymerase 1, and this basically removes RNA nucleotides at primer from the 5' end and replaces them with DNA nucleotides added to the 3' end. And we'll definitely explain this 3' and 5' confusion in the next slide. But essentially, you don't want these primers to be sitting around randomly, especially in the lagging strand when we have a lot of them. So DNA polymerase 3 just removes them and replaces them with DNA. And then we finally have something called DNA ligase, and this basically joins the Ozaki fragments because lagging strand synthesizes via like these Ozaki fragments called here, and they just like kind of go in this direction in fragments, and this just helps join the gaps between them. And so now we're gonna get into the difference between leading versus lagging strand. So this is say your typical double helix double line DNA structure, right? And this dot would represent the origin of replication. So it would open, and then in like this box over here, this is called the leading strand. And so in the leading strand, DNA polymerase 3 alpha RNA primer can continuously synthesize DNA all the way through. As helicase is unwinding, it can synthesize DNA all the way through. And why it can do this is because you guys just need to memorize this, but DNA, these bases, these like little T-shaped things can only be added on the 3' end. And also DNA is like, it's kind of anti-parallel. So therefore, if in this top DNA strand, the 5' end is here and the 3' end is here, so the newly replicated DNA strand, the 5' would be here and the 3' would be here. So over here, this DNA polymerase 3 can effectively add DNA bases here like continuously. And the reason why the other side is called why the other side is called the lagging strand is because remember it can only add in the 3' direction. So if this is the 3', which is basically this one over here, if this is 3' and this is the 5', which is it is, that means its DNA replication is other strand, that is the newly copied strand, the 3' end would be on this side. So it'd be over here, and the 5' end would be over here. And the problem there is that it cannot synthesize continuously like the leading strand, right? It cannot continuously go in this direction. And so it can only go in the opposite direction. But the overall directions of replication still need to stay here. So how does it do that? Well, it essentially goes in like this pattern where it just starts here, synthesizes a bit there, and then moves over here and does this kind of pattern. And this pattern and each of these chunks in the pattern are called Ozaki fragments. And so this diagram really visualizes this. So we have RNA primase adding the 3' end, and then we have DNA polymerase 3 kind of adding in this direction. And then it jumps apart, it detaches, and then attaches to the next RNA primase, and it adds in this line. And then the DNA polymerase 1 just replaces the RNA with DNA, like we said before, and then the DNA ligase tries to reform the bonds there. And then now putting this all together into a bigger picture. So here, just kind of a review as well. Here we have the helicase. It's unwinding the DNA. And as it's unwinding, DNA polymerase 3 in the leading strand continually synthesizes DNA from the RNA primase. So it just continually synthesizes this. And remember those single binding proteins I talked about? Well, it's shown here in the diagram. It just helps stabilize these until this RNA polymerase 3 comes through. And then we have a lagging strand, and this is where we have an RNA primates, just synthesizing these RNA start like places for the RNA polymerase 3 to attach on. So it adds like it added one here, it added one here, and it added one here. And then the DNA polymerase 3 can like add in this direction, add in this direction, and then add in this direction, and then add in this direction. So although the DNA polymerase 3 is kind of synthesizing DNA in the opposite direction, the overall like replication of DNA is still in like this towards the helicase protein's direction, right? And then here we can also see our other proteins in play. As I explained before, DNA polymerase 1 kind of replacing the RNA with DNA nucleotides. And then the lagging strands synthesized in short Ozaki fragments are joined by the DNA ligase. And so that was a very kind of detailed and maybe a bit complicated discussion, but I suggest you guys probably search up on YouTube maybe after this class, like to search up DNA replication and how it works, and you guys may be able to get a little bit better understanding from my explaining, because I can only explain with pictures. And so next we go through mitosis. So remember the cell cycle? After all the other phases, like the G1, S phase, G2, those are all called interphase. But when the cell needs to divide, it goes through a process called mitosis. And this is the division of cells. And a brief overview can be seen here, where one cell basically turns into two cells. And we'll explore how that works in each step. The first step is prophase. So here we have our normal cell coming in from the G2 phase, or just from the normal phase. And what happens in prophase is that the chromosomes condense and they become kind of visible under a microscope distinctively. As you can see here, these chromosomes condense, and the nuclear envelope, remember the cell has a nucleus inside of it, it starts to kind of dissolve and break down. And spindle fibers form and centrosomes move to poles. So this is just, it's not really an organelle, but it's a structure inside a cell that really plays a really important role in mitosis, and we'll see that later. But essentially, this one duplicates in prophase, and it just it kind of migrates and moves to the opposite ends of the cell. And so the prophase sets up for metaphase. And this is where the chromosomes align at the center of the cell. And remember these spindle structures, they form fibers that attach to the chromosomes. And then there's also a checkpoint to ensure proper chromosome separation, which happens in the anaphase, where the spindle fibers are shortened, and that kind of pulls these chromosomes, or the separate chromatids apart from each other, and then they move to the opposite poles of the cells. And then through telophase, they kind of reach the opposite sides of the cell. A nuclear envelope reforms around each set, and then the chromosomes begin to uncoil. So as you can see, two cells are kind of forming right now. And then through a process called cytokinesis, which is basically the cytoplasm dividing into two, the cell membrane pinches in and forms two cells. Or in plant cells, cytoplasm divides, but it doesn't necessarily go through cell membrane pinching. In plant cells, a cell wall kind of just forms in between the two. And then also two daughter cells are formed. And I just like to touch upon the cytokinesis differences in animal versus plant cell. So animal cell, contractile rings, you already know this, but essentially a cleavage furrow forms and it pinches the cell in two. But in plant cells, after everything's set up through like the same stages as animal cell, and we have these two sets of nucleuses basically now, the cell membrane is actually surrounded by a cell wall in plant cells, so it can't really pinch off. So instead, Golgi vesicles travel to the center of the cell and they form a wall, and this creates two daughter cells in the plant cells. So you can see there's differences. And an example of the importance of mitosis is, for example, in plant development. So in a seed, it starts off as a zygote, and then it will undergo mitosis to form two cells, and then this top cell will start to form more and more cells, and this is all through mitosis. And then as the plant grows, its growth is also through mitosis as well. So this really shows how important mitosis is to many organisms. Next, we'll go through chemical communication. So how do cells communicate with each other. The main way that cells communicate through each other long distance is through things called hormones. And these hormones are released by various different organs in your body, including something like the pancreas, your pituitary glands, which are near the bottom of your brain, and your thyroid glands and stuff like that. And they release hormones inside your bloodstream that travel throughout your entire body. And these hormones get distributed and they attach to cells, and that creates a response. So some short examples of hormones are things like melatonin, which helps you sleep, adrenaline, which is famous for the fight or flight response, and then we have the pituitary gland, which helps with growth and development. And hormones are great when the body needs a long, gradual discharge or signaling or a full body response. So it's a great way to get your entire body involved because your blood circulates throughout your entire body. And so melatonin is a type of hormone, and it's a sort of medium-term hormone, we could call it, because its duration is usually only within the 24-hour cycle, and it controls our sleep-wake cycle. So at noon, your melatonin levels are pretty low. But then when in the evening at around 8 or 9 o'clock, for most people on average, melatonin starts being produced by your body. And then it climbs a lot until melatonin peaks in the middle of the night, and then it drops really quickly in the morning, and then you're awake again. And so melatonin is also responding to light. And so melatonin controls your 24-hour cycle, right? But like say you travel to the opposite side of the world to Asia or to somewhere else, and your entire body system or your melatonin levels have to change, right? And so if melatonin, this molecule was just built into your body to be released like every so hours or so, then you won't be able to adjust to this different day and night schedule in the different part of the world, right? And so melatonin is also released gradually to light levels. So light levels kind of trigger the decrease in melatonin in the morning. And this is why I really suggest you guys to, if you guys can, go outside in the morning and take a walk and really let your eyes soak in those wavelengths of light. And that helps you wake up better, and it really helps you focus if you are going to study or do anything else later in the day. So that's what I do. I used to just stay inside after I wake up, but I found it really helpful to actually go outside, take a walk, a very short walk outside, and really try to wake up your body so it's prepared for the day. Next, we have growth hormone. So this is a sort of long-term hormone that accumulates throughout most of your life, and it stimulates growth, muscle, metabolism, and it's actually released primarily during sleep, and it's crucial for growth in children and tissue maintenance in adults. And then we have adrenaline. You guys probably heard of adrenaline. It's also scientifically named norepinephrine. You guys might have heard that as well. But it's famous for your fight or flight response. So essentially when you see something, let's say you see like a tiger over here, right? Your brain, your neural circuits will recognize this. And then it will tell the pituitary gland to release something called ACTH. And then that basically tells your adrenal glands, which sit on top of your kidneys, to release cortisol and adrenaline. So cortisol does other things, but adrenaline does mainly these things. So it tells the liver to convert glycogen to glucose. And we explored this before, but essentially glycogen is the stored form of glucose. And glucose is energy for you to do any activities like run, work, or read, or whatever, right? And so by telling the liver to convert glycogen to glucose, it's basically telling your body to provide more energy to the cells. And so you can fight or flight. It also increases blood pressure due to the accelerated heart rate, and then that corresponds directly to faster breathing, which we'll explore when we get to the circulatory system in the future. It also increases sweating because when you are when your body is consuming more glucose, when it's either running or like fighting, you get really warm and you get really hot, and so increased sweating helps maintain homeostasis. I think we talked about this before as well. And this increased sweating allows your body to get rid of excess heat. It also kind of zones in your vision to become a kind of tunnel vision through dilated pupils. So this helps you focus in on the thing you're trying to run away from or fight. And another important thing is it slows down things like digestion or maybe something else that's not very important to you right now, and you don't want that taking away from your energy. Another thing that happens, it's not directly mentioned here, but that your blood vessels along your important muscles, like your leg muscles or arm muscles, are actually widened to allow more glucose to flow to those cells, those muscle cells that are in need. The next way of cellular communication, and this happens between neurons in your brain, are called neurotransmitters. So neurotransmitters are chemicals that transmit signals between neurons, influencing brain functions like mood, memory, and movement. So this is a simple nerve cell, and we'll explore this when we get to the nervous system. But essentially, the nerve cell has these endings, and they communicate each other through electrical signals. But but neurons are not like they're not like all like physically connected, and the problem with that is that so that electricity can't like pass through multiple neurons all at once, right? And so at these things called gap junctions shown through here is where neurons kind of like touch. Well, they don't necessarily touch because there's still space between them, but how they communicate between each other, because electrical signals can't pass through, is they use things called neurotransmitters. And essentially, when electricity signal passes through here, well, say this neuron just releases some hormones, or well, not hormones. This it releases a chemical signal, and then it attaches to the receptors of the neighboring nerve cell, and then that continues the impulse, and that allows you to like move or think. And dopamine is a very important neurotransmitter. You guys might have heard the word dopamine before, and it's usually classified as the molecule of pleasure, of motivation, or pursuit, and allows you to be happy. A lack of it uh, maybe sometimes can give things like depression or anxiety. And it motivates you to learn. It motivates you to learn new things like skateboarding or reading a book. And also motivates your desire to eat. And I would like to go in a more deep discussion of dopamine during our nervous system unit because I find it's a very interesting thing in your body that you can help utilize to actually improve like your everyday learning and your performance at school as well. So you guys can stay tuned for that lesson because it's more of a practical lesson that you may be able to apply through your everyday life. And the next thing we're going to talk about are gap junctions in cells. So this is another way for cells to communicate. And I think we mentioned this in the first unit, but essentially gap junctions are holes, you could say, in the cells that are touching each other and allows for direct cell-to-cell communication. And this is extremely important for actions like heart muscle because they need to coordinate together to pump blood throughout your body. And by using gap junctions, they can effectively communicate and beat at the correct rhythm, or else you will die because your heart will beat at like an unregulated rhythm and they won't be able to cooperate, you could say, and therefore that's not very good. And so this is like a graph to show the different levels of chemical signaling in your body. So we didn't mention this one, but I'll mention it now. It's something called autocrine, and this is less common, but it's essentially when a cell produces something that targets itself. For example, T cells, which is a part of your immune system, which we'll explore again in the immune system class. But essentially, T cells sometimes they produce hormones that create more of the hormones through like this cycle over here. And this is an example of an autocrine form of chemical signaling. And then there's things called signaling across gap junctions, like we mentioned before. Paracrine, which is when a cell targets a nearby cell. And this is an example of a neurotransmitter. We talked about that between different neuron cells. And then there's something called the endocrine. And this is um, basically when a cell targets a distant cell through the bloodstream. So this is your hormones like adrenaline or your growth hormone or your melatonin. And so next we'll go over the process of meiosis. So not to be confused with mitosis, but this is the reproductive division of cells. So this is this only happens in your reproductive organs, and this basically allows a bunch of organisms in the biological field to reproduce. And it basically produces these cells that then can combine with another cell to form back into something called a zygote and back to this cell. So this process is much longer than mitosis and is split into two main sections. So it has the same stages as mitosis, like prophase, anaphase, metaphase, telophase, but it's split into like prophase 1, prophase 2, and things like that. And we'll explore that right now. And so prophase 1, um, basically, it's the same as mitosis, but except homologous chromosomes pair up. So what do I mean by homologous? Well, homologous chromosomes are chromosomes that come one from one parent and one from another parent. So they essentially have the same genes on them, but they might have different like types of I don't know how to explain this because we haven't went through this, but we'll definitely go through it in our genetics lesson. But essentially, yeah, all you guys need to know is that homologous chromosomes are one comes from one parent and another comes from the other parent. They're pretty similar, so they pair up. And this is also where something called crossing over happens. And we'll explore that in the next couple slides. And then spindle fibers start to form and start to migrate, just like in mitosis. Oh yeah, and this is the slide for homologous chromosomes. So the pairs of chromosomes with the same genes, but possibly different versions. So one from each parent. So these would be homologous chromosomes, and these wouldn't be homologous chromosomes. And this is just a human genome. So we have 22 pairs, 23 pairs, including your XY genes. So we have 23 pairs of chromosomes. And you can see we have 46 total chromosomes if we multiply by 2. And the reason we have 23 pairs is that because each one, for example, this one, uh, they are both, one comes from one parent and the other comes from the other parent. And so they would have the same types of genes on there. Okay, let's oversimplify it and say number 2, this number 2 chromosome pair, these two both encode for, say, eye color, right? But something different might be that this one from one parent might encode for a green eye color, or this one might encode for a brown eye color. So they're the same type of chromosome, but they might have like different traits associated with them, if that makes sense. And these XY chromosomes are chromosomes that identify birth, if you just want to, I mean gender, if you want to know that. So if you have a set of two X chromosomes, you're biologically male. And if you have an X chromosome and if you have two X chromosomes, you're biologically female. And you have an X chromosome and a Y chromosome, you are biologically male. And so this is crossing over. So essentially remember when the homologous chromosomes pair up, something can happen at a place called a charisma, which basically they kind of overlap and then they exchange genetic material. And this basically creates more genetic diversity. And genetic diversity is very important in biology. And we'll explore that in the last couple lessons of this class when we talk about ecology and all that stuff. And we'll explain why genetic diversity is important and why organisms evolved to have this crossing over happen. It's not like it's a random process, but it's not like something that came out of nowhere. There was a reason why organisms evolved to have this type of function. And then next we move on to metaphase 1. Basically, they pair up at the center and spindle fibers attach. And the random pairing also increases diversity, which is important. And this is kind of similar to mitosis, except in anaphase, when they're pulled apart. Remember, in mitosis and anaphase, the individual sister chromatids are pulled apart. However, in metaphase 1, in the anaphase 1, these chromosomes, they're still like attached to the centromere, right? Only like these spindle fibers are pulling apart chromosomes. They're not pulling apart chromatids compared to mitosis. And next we have telophase 1, which is pretty much the same as mitosis telophase. it basically just you can see two like distinct cells kind of forming already, and nuclear membrane starts reforming, and then it undergoes cytokinesis and the cell divides. And so next, this is where it gets kind of interesting for meiosis. We get into something called prophase 2. So now there's two cells, and meiosis forms four cells in the end. So these two cells will undergo a process very similar to mitosis. And so in prophase 2, they condense, the chromosomes condense, and the nuclear membrane breaks down again. And then they will line up at the center, and the spindles will attach. And then they will be pulled apart, very similar to mitosis. The sister chromatids will be pulled apart by the spindle fibers as they shorten. And then eventually telophase 2 forms four cells that are, we call them haploid cells. And the difference between haploid and diploid is a haploid is one copy of each chromosome, and diploid is two copies of each chromosome. So remember, the two copies of each chromosome would be like the homologous pair, right? So what diploid refers to is that you have a homologous pair of each chromosome. And haploid means you'll have one copy of each chromosome. So just an overview of this, I wanted to tell you to keep track of where haploid and diploid cells are. So in this cell, you guys can think about it, but essentially it's a diploid cell because remember, these are homologous chromosomes. So that means you have one from each pair. So this is still diploid. And then in this stage, it's still diploid because you still have these two homologous chromosomes. But unlike mitosis, where everything stays diploid throughout the entire cycle of it, in meiosis, these homologous chromosomes actually separate. And so now these cells are haploids. They only have one copy from each parent. And so this haploid continues till they form four identical haploid smaller cells. And why does this process even happen? Because when organisms reproduce, they need to form this cell. Because when they reproduce, these one from each parent, they will come together and they will form your regular typical cell that you can see here. So the reason you can't or organisms can't use mitosis to create reproductive cells that can then reproduce into a normal cell is that because they're both diploid. And if you have two diploid cells coming together, then you form a quadruploid or something like that. And that's not very good. And that will cause a bunch of issues, definitely. And so they want to kind of turn a normal cell through a process similar to mitosis, but it's not mitosis. It's called meiosis, and it basically forms haploid cells, which then can fuse together through fertilization to form a diploid cell. And that concludes my presentation for today. Do you guys have any questions? If you guys have any questions, feel free to send me a message or send me an email, and I will likely to respond to you in like a day or two. And next, I have a Blooket set up, so we all can play some Blooket. And just give me a quick second to set that up. Guys, what game modes do you want to play? Um, you guys can type in the chat if you guys want to play any specific game modes, or just unmute yourself. Tell me what game modes you'd like to play. Gold Quest? Does anyone else have any other suggestions? I'll give you guys like a minute or two. Yeah, I personally like Monster Brawl as well, but which one is the hardest? I don't have, there's no really like hard, like, yeah, there's like a difficulty scale. My bad. We can check. Crypto hacks, normal, fishing frenzy, simple. This one is for plus, which I don't have, but it's normal. Tower defense, yeah, tower defense is also pretty fun. It's pretty complex. There's a bit more strategy involved. So I feel like this one might be more fun. Monster Brawl is kind of normal. This one is one of my favorites. It's pretty fun. Sip of Dinodes is normal. Battle Royale is very simple. Monster Brawl sucks. I don't know. I just like these things. Tower defense is complex. Cafe normal. Factory is complex. Yeah, factory is kind of weird. It's pretty weird. And then racing is simple. Classic is pretty simple. Book rush is normal. Cafe. So what do you guys want to play? You want to play the most complex ones? You want to play tower defense or do you want to play like cafe or something? Complex. Um. Oh, can I mute the music? There's music playing? Oh, that's very interesting. Let me just mute the tab. Um, mute site. Oh, okay. When I'm in the game. Well, I just muted the site, so shouldn't be a problem. Let's try Tower Defense 2. If it's kind of weird, then just let me know and we can change it. We have some time as well, so okay. So game ends after. Let's do some time. Let's play for yeah, 7 minutes for now. Can we play for 90? 90 minutes? I don't think we have 90 minutes. Like, all right. We'll try 7 minutes right now. Okay, they definitely released new maps last time I played. I don't know what you guys want to play. I guess we should just go for Sunny Meadow. We can just try a normal one. And let's go normal difficulty selection. Yeah. Nightmare mode. Let's try normal first, and then we can see what nightmare mode is about. Yeah, 7 minutes for now. We can host it. Okay, yeah, I'm gonna mute that. Okay. I'll copy the code in the chat as well. So I also posted like the join link in the chat just in case if some people don't know how to access it. I'll give another minute or two for everyone to join in. Yeah, I'm gonna start the game because of time reasons, and also you're allowed late joining. So I was gonna post like, yeah, I'm really good at the Flappy Bird game. Let's post the links again, just in case, and let's start the game. Hopefully the questions are correct and stuff like that. Oh, now you want me to turn on the music? Yeah, the music's on. Can't hear the music? Uh, it's playing in my ear, so I'm not sure. Yeah, I don't know. Don't know how it's gonna work, but. All right. Um, hopefully, hopefully that was pretty fun. Um, do you guys wanna run like a short maybe a simpler game like Gold Quest? All right. Let's run a Gold Quest game. Ooh. Sleep for 5 minutes. Ugh, 5, 5. Okay. Okay. Yo, it might go a bit over time, so if you guys like really need to leave, when it hits that like uh 8 p.m. mark, then you guys are free to like leave if you want. Yeah, I'm gonna start now, and late joining is allowed. So if you guys, oh, you guys can like stay around for later if you want. But like, if you need to leave, then you can leave. All right, let's start. It's close. It's like very close. 4, 3, 2, 1. Oh, it was very close at the end. A lot of things happened. Very close in the end. All right, well, class is over, so you guys can leave if you want. And uh I think next class we'll be going over energy and cells. So we're gonna be going over things like your cellular respiration and photosynthesis. And that's gonna be pretty interesting. So yeah, feel free to leave. Thank you guys for attending, and I'll see you next week. Have a good day.